this is Chapter 21 review. This chapter includes non-fermenting and miscellaneous gram-negative. So there are a few kind of random species that we're going to talk about that are included in these non-fermenters that are of clinical importance. Most of them anyway. So let's just start with some general information about non-fermenters. So most non-fermenters can either be an oxidizer or a non-oxidizer. They grow better aerobically or anaerobically. Most are oxidase positive. Non-fermenters are usually ubiquitous in the environment. They frequently resist reagents or agents such as chlorhexidine and quaternary ammonium compounds. Clinical infections with non-fermenters account for about 15% of all gram-negative rod infections isolated from clinical specimens. Risks involved are immunosuppression, trauma, foreign body implants, and contaminated infused fluids. Infected patients are usually hospitalized or recently discharged from a hospital. So a lot of times this is considered, or these are implicated with nosocomial infections. So some biochemical characteristics of non-fermenters. In a TSI slant, usually there's no change in the butt or the slant, so you'll have a K over K or alkaline over alkaline in that TSI. On the OF or oxidation fermentation tubes, you'll only have acid production in the aerobic tube of the oxidizers and no reaction in that anaerobic tube. A non-oxidizer non-fermenter will show no reaction in either of those OF tubes. Non-fermenter clinical lab clues. So there are 5 primary clues that can help you identify a non-fermenter. In the gram stain, they're long, thin gram-negative rods or coccobacilli. Like I said before, they're generally oxidase positive. They're non-reactive in 24-hour systems used for identifying Enterobacteriaceae species because they're non-fermenters. They're non-reactive on that TSI slant or a KIA slant. And these tend to be resistant to multiple antibiotics. So non-fermenters can be categorized into or organized into smaller groups based on reactions to 3 common tests. So the non-fermenters that I'm talking about that are included in this chapter are Pseudomonads, Acinetobacter species, Stenotrophomonas multifila, Burkholderia species, Flavobacterium and Chrysobacterium, Alkaligenes and Achromobacter, Chromobacterium violaceum, and Sphingomonas species. So those 3 common tests that can organize these into smaller groups are McConkey-Auger reaction or growth. So McConkey-Auger selects for gram-negative enterics and differentiate based on lactose fermentation, the oxidase reaction, and the glucose oxidation fermentation test are those OF tubes. So remember those 3 common tests. So the first group of non-fermenters that I'm going to talk about and is in your PowerPoints are the Pseudomonads. So some general characteristics about Pseudomonads are they're gram-negative bacilli or coccobacilli. They're motile with polar tufts of flagella. They're catalase and oxidase positive. They usually grow on the McConkey and EMB auger as a slow fermenter. So they're not a true fermenter, they're a slow fermenter. They usually oxidize carbohydrates, and some are asaccharitic though. And they tend to have a spreading growth across the agar. Pseudomonids can be split into two groups based on fluorescence activity. So the first group is a fluorescent group. This includes Pseudomonas aeruginosa, Pseudomonas fluorescens, and Pseudomonas putida. And then the second group is the non-fluorescent group, and this includes Pseudomonas suitseri, Pseudomonas mendocina, Pseudomonas pseudoalcaligenes, and Pseudomonas alcaligenes. So of those 7 species, let's first talk about Pseudomonas aeruginosa. So this is the fluorescent group. This most commonly reported isolate of the genus Pseudomonas. It's not a normal flora, but it does account for 5 to 15% of all nosocomial infections. It can cause pulmonary disease in patients with cystic fibrosis. It can cause skin infections after burns. It also is related to something called hot tub syndrome, which is a skin infection. It accounts for 6% of all bacteremias and up to 75% of nosocomial bacteremias. Pseudomonas aeruginosa is beta hemolytic on blood agar. It tends to have a fish scale appearance as it spreads across the agar. It's gram-negative bacilli. Some do have a green hue due to a pigment called pyocyanin. Most strains produce a fluorescent pigment called pyoviridin. So know the difference between those two terms, pyocyanin and pyoviridin. Many strains produce a fruity odor, a grape-like fruity odor. They can grow at 42 degrees Celsius. They are cetramide positive on acetramide agar and acetamide positive as well. I believe that is all the facts I want you to know for Pseudomonas. The next species is Pseudomonas fluorescens, and this is the second one in the fluorescent group. And the third one is Pseudomonas putida. So these members of that fluorescent group differ from Pseudomonas aeruginosa in that they do not produce pyocyanin and they do not grow at 42 degrees Celsius. So know that. These two species can be differentiated by gelatin hydrolysis. So that's a test we can use to differentiate between these two particular species, fluorescens and putida. So let's go to the non-fluorescent Pseudomonas group, which had 4 organisms. That was Pseudomonas suitseri, and this has a characteristic wrinkled, leathery colony appearance with a brown pigment. Number 5 was Pseudomonas mendocina, and that's rarely isolated from humans. It's usually a contaminant. And then your 6 and 7, or your 3rd and 4th species in the non-fluorescent Pseudomonas group is Pseudomonas pseudoalcaligenes and Pseudomonas alcaligenes, and these are usually contaminants. So the next group of organisms in this non-lactose fermenting group in miscellaneous gram-negatives chapter is Acinetobacter species. There are 2 species that are most common, and that is Acinetobacter baumannii and Acinetobacter iowaphii. So these are usually seen in nosocomial infections associated with ventilators, humidifiers, catheters, and other devices. And as many as 45% of patients with tracheostomies may be colonized with this particular, or these 2 particular organisms, one of the 2. Acinetobacter is second only to P. aeruginosa in frequency of non-fermenters frequency of isolation of non-fermenters in the clinical microbiology lab. There are multiple drug-resistant strains of Acinetobacter baumannii, and these drug-resistant strains are seen in several Iraqi war veterans. So know that little fact or detail. Some identifying characteristics on the agar in the gram stain. They are gram-negative rods, but they can appear as gram-positive cocci on smears from blood culture bottles. So that's kind of unique to this group of organisms. They're catalase positive, oxidase negative. Some species produce a purplish color on MacConkey plates, which can be mistaken or make it appear as though it's a lactose fermentation positive organism, but it's not. So moving along, the next group of organisms, or one particular organism that's mentioned in this chapter, is Streptophomonas maltophilia. That's a tough one. Streptophomonas maltophilia. This is the third most common isolated non-fermentive gram-negative rod in the clinical lab. Fairly ubiquitous in the environment in hospital settings. It's not part of the normal flora, and it is the single most important risk factor associated with this organism is the presence of a venous catheter. So let's move on. We've got 3 Burkholderia species. So in the Burkholderia group, there's 3 species we'll talk about, and that's Burkholderia cepacia, and this particular organism is capsulated, most often associated with pneumonia and cystic fibrosis patients. It's oxidase negative and very difficult to treat or get rid of. So that's cepacia. The second Burkholderia species is Burkholderia mallei, or mallee. This causes glanders. It's a zoonotic infection primarily affecting livestock, such as horses, mules, and donkeys. Mucous membranes of the nostrils can become infected, and also there's an associated swelling of the jaw. This particular organism, Burkholderia mallei, is considered a Category B bioterrorism agent by the CDC. It's a very, very rare. I think only 1 case of glanders has been reported in the U.S. in the last 50 years, and it was lab acquired, so it's very rare. The third species of Burkholderia is Burkholderia pseudomallei. This organism causes melioidosis, which is an aggressive granulomatous pulmonary disease. And with this disease, you have development of lesions in your lungs. It's found frequently in Southeast Asia, Northern Australia, and Mexico. Not common here in the U.S. Pseudomallei produces non-fermentative wrinkled colonies on blood agar, and they tend to have this earthy odor. So that's your 3 Burkholderia species. So there's a next small little group worth mentioning, and that's Flavobacterium and Chryssobacterium. These are clinically important species. They're oxidase negative and indole positive. They produce a yellow pigment and also have a fruity odor like Pseudomonas aeruginosa. Most diseases caused by Flavobacterium species is this particular organism. That's Flavobacterium meningosepticum. So know that one organism is the causative agent of disease by this. The next group, or 2 groups in this chapter of miscellaneous gram-negatives is alkaligenes and Achromobacter. Of the alkaligenes, the most commonly isolated species are Alkaligenes faecalis, and of the Achromobacter group, it's the most commonly isolated is Achromobacter xylosoxidans species, or xylosoxidans, subspecies xylosoxidans is in particular. They can grow in water. They're resistant to disinfectants, such as chlorhexidine and ammonium compounds, or quaternary ammonium compounds. All species are oxidase positive, and the Peritrichus flagella make them motile. They usually grow well on most lab media, including your MacConkey agar. And all species reduce nitrate to nitrites, so they're nitrate positive. So there's two more groups in this chapter. The next one is Chromobacterium violaceum. This species is a gram-negative rod that may be oxidase positive, and it may appear as curved rods on a gram stain. This organism produces something very unique. It's a very pretty appearance on the chocolate agar plate. It produces a violet pigment about 91% of the time. And this violet pigment is called violacin. So know that that characteristic is unique to Chromobacterium violaceum. It's usually an opportunistic pathogen. Most infections are seen in immunocompromised patients. And so that leads us to the very last little group in this gram-negative miscellaneous chapter. That's Sphingomonas. The most common species of Sphingomonas seen in the lab is Sphingomonas paucimobilis, and it can be isolated from water as well, including swimming pools and whirlpools. So know that Sphingomonas can be found in those two types of places. There's a yellow pigment present, and it does not grow on MacConkey agar, so that's kind of unique to the Sphingomonas. And these are weakly oxidase positive and indole negative. So that's all you really need to know about those. There's a little bit about each one of these groups that you need to know, just to be familiar with them, that it's possible to see these in the laboratory. And just be familiar. They're out there. You may come across them. So anyway, that wraps up Chapter 21. Study your PowerPoints and highlight all those things I've mentioned, and that's that.